Hi, my name is Mr. Kosis, and today we're going to be talking about the processes of mitosis and meiosis. Now, before we jump into them, first let's talk about the checkpoints that the cell must go through before it can go through these processes. The first of which is G1, in which cell growth occurs. The second, the S phase, chromosomes are duplicated so that there are two sets of 23 chromosomes. And the final stage, G2, ensures that there is no error before the cell is duplicated. Mitosis. The purpose of mitosis is for cell reproduction, regeneration, and growth. In the first phase, prophase, chromatin fibers become more tightly coiled, and nuclei begins to disappear. Centrosomes move away from one another, and mitonic spindle begins to form. In the second phase, prometaphase, microtubules from the centrosomes invade the nuclear area, and chromosomes condense. Each of the two chromatids now have a kinetochore, a specialized protein structure, and microtubules attached to the kinetochores, pulling chromosomes back and forth, and non-kinetochores interact with those of the opposite poles. In metaphase, chromosomes move to a metaphase plate, an area that is an equal distance from each chromosome, or centrosome. Chromosomes attached to the centrochore, microtubules from each pole. In anaphase, cohesion proteins holding the chromatids together are cleaved, allowing them to separate. In telophase and cytokinesis, two daughter nuclei form in the cell, and the nuclear envelopes form. Chromosomes become less dense, and the remaining spindle depolymerize. Cleavage furrow forms, and pinching the cell into two. In the process of meiosis, we reduce diploid cells to haploid cells called gametes. This is a fairly similar process to mitosis. In prophase, homologs become physically connected to each other by synapses, and crossing over occurs, the genetic rearrangement between non-sister chromatids, exchanging corresponding segments of DNA molecules. In metaphase 1, homologous chromosome pairs are lined up at the metaphase plate, and chromatids of one homolog attached to kinetochore microtubules from one pole, and vice versa. In anaphase 1, homologs separate due to the breakdown of proteins responsible for holding them together, and move towards opposite poles guided by the spindle. In telophase 1 and cytokinesis, each half of the cell has a completed haploid set of duplicated chromosomes, and cytokinesis, two haploid daughter cells form. In prophase 2, spindle apparatus forms, and chromosomes still composed of two chromatids at centrosomes move towards the metaphase 2 plate. In metaphase 2, chromosomes situated at the metaphase plate, due to crossing over in meiosis 1, In metaphase 2, chromosomes are situated at the metaphase place. Due to crossing over in meiosis 1, the two sister chromatids are not identical. The kinetochores of sister chromatids are attached to microtubules extending from opposite poles. In anaphase 2, breakdown of proteins holding sister chromatids together. Chromatids move towards opposite poles. The last and final stage of meiosis is telophase 2 and cytokinesis. Nuclei form during telophase 2 and chromosomes decondense. This process of meiosis creates 4 daughter cells from one parent cell, each with a haploid set of unduplicated chromosomes. The 4 daughter cells are genetically distinct from one another and the parent cell.